Okay, should I start or should I wait for more people? Hello, everyone. My name is Farah, and I'll be talking about renal histology today. I'll be giving you this crash course. It's going to be a bit heavy today, but if you guys can focus with me, I'm going to make it very simple and easy to understand. Okay, I'll be covering 3 lectures today. The first one being the microscopic histology. It was divided into 2 parts, and the second one is macroscopic histology of the kidneys, which involve the ureters, the bladder, and so on. So let's get started. And you can stop me at any point if you have any questions. All right, so here are the objectives I'll be covering today. There's plenty. It's very overwhelming, but don't worry about that. Like, I'll get all through them very quickly. Okay, so let's start with the organization of the kidney. The kidney, we have 2 kidneys in our body, okay? It is divided into lobules. So we have in each kidney, we have 7 to 18 lobules. These lobules, like you can look at them here. So let me put the pointer. Okay, you can see we have many lobules here. You can see around like 7 here, but we have 7 to 18 in each kidney, okay? And these lobules, they consist of 2 parts, mainly the cortex and the medulla, okay, or the medullary pyramid. We call it the medullary pyramid because it looks like a pyramid, you know, a triangle, okay? Then we have the renal papilla, which is at the end of the renal medulla, and the area criprosa and the minor calyx. I'll get all through them, okay? Okay, the human kidney is the organ that synthesizes urine and secretes it. So here, urine is synthesized, and then it's secreted. We have, as I said, 7 to 18 lobes, and the lobes of the kidney consist from outside to inside. We have the, we have the fibrous brown capsule, then we have the cortex, then we have the medulla. This is a slide of a mouse kidney. So you can only see one lobe here. However, in the human kidney, we have like 7 to 18. And then we have the renal papilla, which is this area right here. Okay, let's talk about it. So zooming into the kidney, we'd find millions of microscopic hollow tubules and ducts. Structure, they have only one layer. Now, why do you think we have one layer? Can anyone answer me? Why do we have one layer for urine formation? Okay, we have one layer because we need molecules to move from, like very easily from the capillaries to the tubules, right? We need one layer because they cannot pass through multiple layers. So they have one layer from all of these tubules. They have the tunica intima only, okay, which consists of a simple epithelium and endothelium, whether that's squamous, cuboidal, or columnar, and a basal membrane. So there's a very high permeability and low protection here. The function is urine synthesis, and it happens where there's exchange with the blood or capillaries, and they're located deeply in the kidney. Okay, so now we know that like macroscopically, so when we have like the larger ducts, we cannot make urine. We can only transport it. Why? Because there's like around 3 layers over there. So urine can only be transported. It cannot be made. Molecules cannot move easily through those layers. Now, to form the macroscopic hollow tubes, we have 3 layers: tunica intima, media, and externa. They consist of the stratified transitional epithelium, and this is later in the urinary system where we have the minor calyx, the ureter, and so on. I'll talk about it later. And then the basement membrane. There's very low permeability here, and there's a high protection. Okay, this is where urine transport and excretion happens to the toilet. These are the kidney and other organs closer to the outside environment. The outside environment being the outside world where, we move the pee to the toilet. Okay, let's talk about the ducts here. This is a very, confusing slide, but I'll try to explain it. So urine formation begins here. This is the glomerulus, and this is the glomerular capsule. Together, they make the renal corpuscle. So this circular organ is called the renal corpuscle, and we have plenty of them in the cortex of the kidney. Here, we have the afferent arteriole, where we need the molecules to move from the blood to the glomerular capsule. So here they start moving. So the green arrows is the movement of the urine from the glomerulus to the collecting ducts and so on. So this is the direction of the urine movement. So urine moves from the glomerulus. So some people might think that the glomerulus is this entire structure. No, it is only these capillaries. So urine moves from the afferent arteriole to make the glomerulus, and then moves into the glomerular capsule. From there, it moves into the proximal convoluted tubule. It's called convoluted because it's like very squiggly and like convoluted, okay? And then this is the pars recta of the proximal tubule, or another name for it is the distal the thick descending limb of the loop of Henle. This entire loop, this loop is called the loop of Henle. We have the thin limbs and the thick limbs, but this is also part of the proximal tubule, you could say. So this is the pars recta, and this is the thin descending limb of loop of Henle. This is the nephron loop. All the way from here, we're talking about the medulla, okay? And everything above that is the cortex. And this is the thin ascending limb of loop of Henle. And this is the thick ascending limb of loop of Henle, or the pars recta of the distal convoluted tubule. And then it moves all to the collecting duct, okay? Okay, now we have 2 million nephrons in our kidneys, and they all have these tubules. So this entire structure is called the nephron. This is the functional or the parenchyma of the kidney. This is the functional. So what is the function of the kidney? It is the synthesis of urine and its transport and excretion. So the functional part of the kidney is the nephron. So like the functional part, for example, of the lungs is the alveoli, where there is blood gas exchange. This is the functional part of the kidney. It's a nephron. Okay, the proximal tubule, we have this convoluted shape, and the distal tubule, and so on. Okay. This is the same slide that I talked about in word form. We have the microscopic or the functional unit of the kidney is the uriniferous tubule, or another name for it is the nephron, but we refer to it as a nephron, okay? And these are the tubules that are one layer thick. This is where urine exchange happens, okay? Or molecule exchange happens, where there's urine and filtrate formation and so on. And yeah. So this is just the cascade of the urine flow. So it moves from the renal corpuscle, which was that circle, okay, which consists of the glomerular capsule and the glomerulus, to the proximal convoluted tubule, then the pars recta, or the thick descending limb of the loop of Henle, then the thin descending limb of the loop of Henle, then the nephron loop, then the thin ascending limb of the loop of Henle, then the pars recta, which is the thick ascending limb of the loop of Henle, then the macula densa of the distal convoluted tubule, which I'll talk about later, okay? And then the distal convoluted tubule, or the pars convoluta of the distal convoluted tubule, and then the collecting duct. From there, this is the nephron. Now then, from the collecting duct, we have the collecting duct system, which is different from the nephron itself. This is where urine isn't there isn't any urine like reabsorption and formation. There's only urine transport, okay? From here, we have the cortical collecting duct. From the name cortical, it's present in the cortex. Then we have the medullary collecting duct. So this you could say this is the cortical collecting duct, then the medullary collecting duct, okay? And then we have the papillary collecting duct, which is all the way down at the papilla right here. So this is the papillary collecting duct of Bellini. We call it the papillary collecting duct of Bellini, okay? And then it moves to the minor calyx, okay? Now, about the papillary collecting ducts of Bellini, we have around 30 papillary ducts per one papilla. So let me show you here. Okay, so all of these are the renal corpuscles, okay? And these are the nephrons. They're showing us only one nephron in this model, okay? But there's around so many. There's so many in one lobe, okay? Around 5,000 of those drain into one renal papilla, or papillary collecting duct, one papillary collecting duct of Bellini. This is the collecting duct, okay, of Bellini. We have around 30 of those. There's only one shown in this model, but if 30 of those join, they drain at the area cribrosa. This is the area cribrosa. Basically, it's cribriform. Cribriform means holes. It has holes. So these papillary collecting ducts, they drain into, so they make holes and at the area of cribrosa, and these holes all drain into the minor calyx, which is not shown here. I'll show you later. Okay. Okay. Let's talk about the nephrons. We have two kinds of nephrons, okay? The nephrons this is the nephron, okay? We have two kinds. First of all, the cortical nephrons, which is this one. The cortical nephron is closer to the renal capsule, and it just dips a little bit into the medulla, whereas the juxtamedullary nephron, this one, these types of nephrons are dipping deeper into the medulla, and these mostly focus on the urine formation plus countercurrent multiplier. A countercurrent multiplier, you learn about this in physiology, but basically it makes a concentration gradient for urine to make a hypertonic interstitium for urine to get reabsorbed and to make, urine more concentrated. But I won't go into this very much because you learn about it in physiology. And you can't really see this on histology anyway. Okay? In the cortical nephron, there's only urine formation. Okay. Then we have the collecting system. So this big arrow here shows us the microscopic tubules of only simple epithelium, whether that's squamous, columnar, or cuboidal. So there's only simple meaning one layer, okay? And as I mentioned earlier, one layer is necessary for molecule exchange. Okay, then we have this is where macroscopic urine flow begins. From the minor calyx to the rest of the world, okay? Here we have macroscopic tubes and stratified epithelium. We have stratified epithelium, not simple, okay? And here where you start seeing the transitional epithelium, okay? So where do we see the junction between simple epithelium and stratified epithelium? Can anyone tell me? Or simple epithelium and transitional epithelium? Anyone can tell me?
The papilla?
Yes, this is what we see at the papilla or the area cribrosa. Yes, you're correct. Okay. So can anyone tell me about how many papillary collecting ducts of Bellini do we have per lobe, that drain into the area of cribrosa? How many drain into the minor calyx? It's 30, yeah. Yeah, correct. Okay. So here you can see the minor calyx, by the way. These are the funnel-shaped parts of the kidney. So this is the papilla, right? And it is draining to the minor calyx that you can't see here. But here is the papilla, and all these papillary collecting ducts, there's around 30, and they drain into the minor calyx. These are the major calyces, okay? And this is the pelvis. But I'll get into those later. So the papillary collecting ducts drain into the minor calyces, and this is the beginning of the macroscopic urine flow, as I mentioned earlier. The microscopic tubules, as I mentioned, only simple epithelium. And the papillary collecting ducts, we have also simple, but it's columnar. Why columnar? Can anyone guess why we have columnar at the collecting duct of Bellini? Why is it like a longer cell than these, the other cells, the cuboidal and squamous? Why do we have longer cells here?
For the reabsorption of the water.
No, we have a longer cell here, or like bigger cells, because there's more urine volume, basically. As I mentioned, all of these nephrons, like 5,000 nephrons, they're all draining into this duct. So it has to carry, like they're all merging and draining into this duct. So it has to carry this big volume of urine. So it needs bigger cells to hold that, Someone asked a question, are the medullary collecting ducts and papillary collecting ducts continuation from the cortical collecting duct? Yes, this is the cortical collecting duct, basically. It just continues into so we just name it differently based on the location, okay? Yeah. These are the collecting ducts here, but then they merge into the papillary collecting duct, okay? The cortical and the medullary, they merge into the papillary. Okay. Now, this is a very overwhelming slide, I know, but we have to know like the types of different cells that we see in the nephron. Okay. Sorry. Okay. So, so here are the different types of cells that you would find in the nephron. Let's begin with the Bowman, the renal corpuscle. We have the glomerulus, and in the glomerulus we have the capillaries and the arterioles. So basically, this is just simple squamous epithelium, endothelium. Okay. But it's covered by podocytes, which I'll talk about later. But basically, these podocytes, which are like modified simple squamous, the podocytes, from the word podo means like feet, so they have like feet or projections that are kissy huggy as Dr. Annie mentioned, kissy huggy the capillaries in the glomerulus. Now here is the Bowman's capsule, or yeah, this is the capsule. It looks like two layers, but it's like folded. So the outer layer of Bowman's capsule is simple squamous. Okay, the inner layer, or the visceral layer, which is in close contact with the arterioles or the glomerulus, this is the modified simple squamous, which is the podocytes. Okay, these are kissy huggy with the capillaries. I'll show you later. Wait, let me show you. I showed, so here you can see, so basically this entire glomerulus should be yellow, okay? Because you have the capillaries, but then they're covered by the visceral epithelium of the Bowman capsule, or the podocytes. These are the podocytes. See, they have little feet like that. And these little feet actually create filtration slits where there's molecule exchange. These slits allow molecules to move freely. So this whole thing should be yellow, but they just didn't cover the entire thing for like for you to realize that there's endothelium underneath that. Okay, then we move on to the proximal convoluted tubule. The proximal convoluted tubule has cuboidal cells, simple cuboidal epithelium with microvilli. This is the very important part about the proximal convoluted tubule, and also the pars recta of the proximal convoluted tubule. This whole area has cuboidal epithelium, simple cube. So this entire nephron is simple, by the way. So this area is simple cuboidal with microvilli. Does anyone know why we would have microvilli in this area? Like, what is the function of microvilli? Or what is the function of the proximal convoluted tubule?
So
It
now
is,
is for the reabsorption.
yes, reabsorption, exactly. So we have many microvilli here for reabsorption. Then moving on to the loop of Henle, this entire area, the thin descending and ascending, and the loop of Henle, we have simple squamous epithelium. And actually it's really thin, so you could imagine simple squamous, yeah. Then we move on to the distal tubule, or including the pars recta of the distal tubule. By the way, recta means erect or standing. And so the pars recta of the distal tubule and the convoluted part of the distal tubule, this entire area is also simple cuboidal, but what do we not have here? We don't have microvilli. So this is how you differentiate the proximal convoluted from the distal convoluted tubule. And I'll show you histology slides that show the very clear difference between those later on. Then we have the cortical collecting duct, the medullary collecting duct, the papillary collecting duct right at the bottom. These contain simple cuboidal epithelium, but in the cortical collecting duct, we have two types of cells, and in the rest of it, we have one type. So in the collecting ducts, we have some cells that are called principal cells. These cells are light colored, they're light cells. And most of the entire thing, we have principal cells or light cells. And then in the cortical portion of the collecting duct, we would have these cells that are called intercalated cells or dark cells. So you'd have many principal cells and only like, a few, like haphazardly placed dark intercalated cells, okay? And in the medullary and the papillary collecting ducts, we only have principal cells or the light cells. In the cortical and the medullary, they're cuboidal. Yes, in this area. And then when you move on to the papillary area, it is columnar. Okay, they're longer. Okay. Okay, moving on. This is basically what I said already. They're simple cuboidal in the cortical collecting duct and the medullary collecting duct. In the papillary, they're columnar, and you have a short microvilli here. Okay, yeah, only principal cells here, and principal and intercalated cells here. Now, these sometimes they're referred to, so the intercalated cells are sometimes referred to as dark cells, and principal cells are sometimes referred to as light cells. But when Dr. Annie asks you to mention which type of cell, don't say dark or light. Say principal or intercalated. Use their longer names, okay? Their real names. This is basically what I said already in word form if you don't like pictures. The whole nephron is made of simple epithelium. The stratified or transitional epithelium begins at the minor calyces, okay? This is just a summary slide for your reference. Okay, let's start with the blood flow of the kidneys. We start with the renal arteries. Renal arteries immediately emerge from the aorta, okay, from the abdominal aorta. And each renal artery goes to one kidney. Okay, renal arteries, they branch into segmental arteries. Then we have in each lobe, first of all, we have the interlobar artery. Interlobar means between the lobes. So, you know, we have like 7 to 18 lobes. So between those lobes, we have the interlobar arteries. Then we have the arcuate arteries. These arteries, from the name arc or arch, they make an arch, okay, between the cortex and the medulla. Then these arcuate arteries, they give us the interlobular arteries. So these lobules in the kidney. I'll explain to you what a lobule of the kidney is, okay? Lobules in the cortex, okay? So these interlobular arteries come from the arcuate arteries. Then, going deeper, we have the interlobular arteries. They give us the afferent arterioles. They branch into so many afferent. It creates, as Dr. Annie mentioned, Christmas tree. So it looks like a Christmas tree. So this is the I guess you could say the tree trunk, and these are the, I don't know, the vines. So these are the afferent arterioles coming out of the interlobular arteries. The afferent arterioles, they move onto the renal corpuscle to make the glomerulus. So they branch into the glomerular capillary bed. However, in the glomerulus, okay, let me show you something. So in the glomerulus, we have, so as we know from CVS or cardiovascular block, when vessels branch, they create, you know, capillaries. They're still like moving red blood or like oxygenated blood. Then when they like merge, they create venules where blood should be, you know, going back to the heart, right? However, here this is an exception, okay? The afferent arteriole, okay, it branches into the glomeruli to make the glomerulus, I guess, creates these capillaries. But when they merge, they don't become blue. They don't make a venule. They make another artery, which is the efferent artery. Now, does anyone know or like have an explanation as to why it becomes an efferent artery and it not like a venule? Like, can anyone guess?
You supply the rest of the
The kidney.
uriniferous artery. Yeah.
Yes, so yeah, you're correct. So the afferent arteriole, because when it makes the glomerulus is used to make the urine and the filtrate and so on. But then we need to supply the actual parenchyma of the kidney, the nephrons and everything else in the kidney. So that's why we have the efferent arteriole, okay? We need to supply these actual nephrons. They need to stay alive. So this is what the function of the efferent arteriole is, okay? So let's go back. So we made the efferent arteriole, then the glomerular. This is the first, that we have two types of capillary beds. The first, or abnormal red to red capillary bed, as you see here. Then we have the efferent arteriole, okay, which is coming out. Then we have the efferent arteriole branches into the blood vessels that, as our colleague mentioned, should supply the nephrons and everything, or the uriniferous tubules. And these are called peritubular capillaries. These are normal capillaries because normal as in when they merge, they create red to blue capillary bed. Basically, they supply oxygen, and then when they merge, we have the deoxygenated blood going into the venules. Okay, and so we have two places where we have the peritubular capillaries in the cortex. These are the peritubular capillaries in the cortex, and then we have them in the medulla as well. In the medulla, they have a special name. They're the same thing, the same job, same types of capillaries, but they have a special name, which is vasa recta. Vasa recta, because they're erect or straight in the medulla. Everything in the medulla is straight. The tubules, the artery, the arterioles, the venules, you know, or the capillaries, everything is straight. So we call it vasa recta. And we also call the venules here. Also call them vasa recta. Okay, and here it's normal red to blue capillary bed. So does everyone understand the difference between the two capillary beds we have? Yes, okay. Okay, now moving on to the venules. The peritubular capillaries, they merge to make the venules, and this goes back to the heart, and inferior vena cava, etc. And then we have just like we had the interlobular artery, we have the interlobular vein. And just like we had the arcuate artery, we have the arcuate vein. And we have the interlobar vein, which is between lobes of the kidney. And then we have the segmental vein, like the segmental artery, and then the renal vein back to the inferior vena cava. So blood flow in the kidney, as I mentioned, is very unique because we have two types of capillary beds. Okay, we have the red to red and the red to blue. Okay. Okay, two functions of both of the types of capillary beds, which is oxygen and nutrient delivery to the nephrons, which is the function of the second normal to capillary bed. And then we have the exchange with nephrons to make urine. This is the function of the red to red or unique capillary bed. Okay, the vasa recta, they supply, they are the straight blood vessels, whether they are capillaries or venules in the medulla. And yeah, they are, so we have arterioles, capillaries, venules in the medulla. They're called vasa recta. Okay. And yeah, I mentioned all of that. Okay, let's just talk about the cascade of the blood flow in the kidneys. So blood comes from the interlobular or the radiate artery, which is number 5 here. Okay, then it branches, makes a Christmas tree to make the afferent arterioles, each one going to a glomerular, renal corpuscle. Okay, these afferent arterioles branch to make the glomerulus, which is where the molecule and the urine filtrate happens. And then they branch to make the glomerular capillary. And then these capillaries merge to make the efferent arteriole, which is coming out. And the efferent arteriole branches into the peritubular capillaries that is supplying the rest of the kidney. Okay, basically this is just a slide showing you how these peritubular capillaries, they're kissy, huggy with the nephrons in the kidney. See the glomerular, so this is the glomerular capillary bed. These are the peritubular capillaries in the cortex. Look how they're like kissy, huggy with all the tubules here in the nephron. And here also in the medulla, same thing. But the special thing about them is that they're called vasa recta because they're, straight. But they're the same exact like type of vessels. Okay. Okay, let's zoom into the cortex. Let's talk about the histology part. Okay, so this is blood flow in the kidney. So first of all, we have the peritubular capillaries here. You see them. Why are they peritubular? Because they're not in the glomerulus. So in the glomerulus, you have the first bed of capillaries, which is the specialized red to red bed covered by podocytes. So how do we identify capillaries? We see the red blood cells or the erythrocytes, all these pink circles. These are erythrocytes. So this is where there's a capillary, okay? Even though you can't really, you might not notice the simple squamous epithelium surrounding it. But if you see red blood cells, oh, there's a capillary right here. So this is the first bed of capillaries in each glomerulus. You can see one glomerulus in this slide, okay? And then we have the peritubular capillaries, whether they are cut longitudinally or in a cross section. So when you have a vessel and you cut it in cross section, you would see a circle. So here, this one was cut in cross section, and these ones were cut longitudinally. Okay, basically these are peritubular capillaries because they're outside of the glomerulus. So anytime you see a capillary that is not in the glomerulus, these are peritubular capillaries. Okay, let's talk about urine formation. So filtration begins at the glomerulus, as I showed you in the nephron. So it begins at the glomerulus, where blood moves from the glomerulus or the capillaries into the Bowman space. This is where filtrate happens. The end product is filtrate. It's not urine yet. It has to move all through those two to like create for it to be called urine. Okay, then we move it. The urine moves to the proximal convoluted tubule. So this is a proximal convoluted tubule, and because it has microvilli, as I mentioned earlier, look, microvilli. So what happens here? Reabsorption. Okay, we have reabsorption here. Then reabsorption. So where is urine going from, or where are the molecules? They're going from the duct to the capillaries or the peritubular capillaries that are kissy kissy, huggy huggy with the tubules. And so here we have the pars recta of the distal convoluted tubule. Here we have secretion because we don't have microvilli. Here we have secretion. Okay, and so secretion means the blood is, so the molecules are, or the urine is going from the capillaries back into the tubules. Okay, secretion or excretion. Okay, let's to like really understand how filtration happens, we have to look at it in the electron microscope for us to understand like how are the molecules moving. Okay, so here's a picture. This is our glomerulus. Okay, so as I mentioned here, filtrate happens from the glomerulus to the Bowman's capsule. Okay, so here let's see. So this is a picture or an electron microscope picture of the basement membrane and so this is basically the one capillary in the glomerulus, okay? And it is surrounded by podocytes, right? As I mentioned earlier. So this is the CVS part or the capillary. And here we have the endothelium, this simple squamous endothelium. It has fenestra, or fenestra means windows. So these windows allow particles to move from the blood through the basement membrane and to the lumen, or EM is basically the lumen of the tubule, or this is the Bowman's space, sorry. Okay, so here, can anyone guess what these like thingies are, these projections? They're on the outside of the capillaries. These are the podocytes, okay? I showed you guys the podocytes earlier. They are covering the capillaries. Podocytes, they have like their feet, they're clinging onto the capillaries around them. These basically, this is the visceral part of the Bowman capsule, okay? These that are covering the capillaries, and they have these filtration slits, okay? You can see them here. These are the podocytes. These are their feet. And the molecules basically just move through them, okay? And we have these tiny, like, I guess, connecting membranes. They're called diaphragms, okay? Urinary diaphragms, okay? This is just another electron microscope picture of the podocytes themselves. See how they look like feet surrounding the capillaries and the glomerulus. Pretty neat. This is another picture showing us all the different types of cells in the glomerulus, starting with the actual red blood cells. See all this pink stuff. I told you, whenever you see pink, a pink circle or whatever, this is the erythrocyte or red blood cells. You know that there's a capillary here. Okay, so these are red blood cells. So if this is red and capillaries have like capillaries are made up of simple squamous epithelium, right? What are all of these purple circles. These are mesangial cells, okay? These, these are mesangial cells and podocytes. So the podocytes here, right? I mentioned number one, okay? Podocytes that are kissy, kissy, huggy, huggy, the capillaries, but we also have mesangial cells. Mesangial cells are the supporting cells of the glomerulus, you could say. Okay, I'll talk about them later. This is number three is the Bowman's space, okay? And this is where urine just moves from Bowman's space to this is the proximal convoluted tubule, okay? And yeah, number five is basically telling you that there are mesangial cells in between these capillaries, in between the glomerulus. So in these spaces here, you would have mesangial cells. And then this is the parietal part of Bowman's capsule, or the outside part of Bowman's capsule, which is simple squamous epithelium, which is here. This is simple squamous. Okay. Okay, there are four kinds of cells in the renal corpuscle, which is the endothelium, or the simple squamous epithelium of the capillary beds in the glomerulus. Then we have erythrocytes inside the capillaries, or red blood cells, that we identified by their pink color. And then we have the podocytes of the urinary system surrounding the capillaries in the capillary bed. And then we have the interglomerular mesangial cells. So we have two kinds of mesangial cells. The interglomerular, from the name, they're inside the glomerulus. And then we have the extraglomerular mesangial cells. But the interglomerular mesangial cells here, we need to focus on them because they have several functions, but their most important function is phagocytosis of trapped molecules. So for example, when you have molecules moving in and out, you know, the molecules moving from the capillary to the Bowman's capsule, you know, and there's a molecule that got stuck or molecules that are not moving, these cells basically phagocytose them to keep the area nice and clean. Okay, they also provide structural support of glomerular capillaries. They have other functions, but these are the two most important, especially the phagocytosis. This is their primary function, the mesangial cells, just to keep area nice and clean, phagocytosis. Then we have stromal cells. So I mentioned that the parenchyma, or the functional part of the kidney, is the nephrons. This is the function of the kidney. And each nephron is performing this function. We also need something to hold all of these structures in place. These are the stroma, or the stromal cells. The stromal cells, we have several. We have the intraglomerular mesangial that I mentioned here. And then we have the extraglomerular, which are called Lacey cells, or Lassus cells. And then we have interstitial cells that are in the cortex, or like in the rest of the kidney. Interstitial cells are mainly fibroblasts. We learned that from foundation, I guess. And they synthesize collagen, type 3 collagen and type 4 collagen. Type 3 is like the more, like, you could say, friable type of collagen, I guess. And it's just to hold and support the nephrons in place. Type 4 collagen is just famous for the basement membrane. Wherever you see a basement membrane, this is type 4 collagen, and it is synthesized by interstitial cells, which are fibroblast-like cells. Okay, I want you to just know two things here. So this is the urinary pole, where the urine flows from the Bowman's capsule to or the filtrate. Actually, it's not urine here because for it to be actual urine, it has to go through the tubules. But basically, the filtrate that was made here flows through this urinary pole and exits at the proximal convoluted tubule, and then moves through the entire nephron. Then we have the vascular pole. The vascular pole is a bit special because it has specialized types of cells here in this triangle. So this triangle is made by the afferent artery, the efferent artery, and the distal convoluted tubule. And this area, we call it the juxtaglomerular apparatus. And I'll tell you what that is. The juxtaglomerular apparatus consists of three things: the juxtaglomerular cells, the macula densa, and the extraglomerular mesangial cells. Okay, this is just, it sounds like, I don't know what she's talking about, right? But I'll explain it to you. So the juxtaglomerular apparatus, basically its main function, like, you have to understand the function so you can understand why these cells are here, basically, or what they are. So it has two functions. Basically, for the blood pressure in the body, like to maintain the homeostasis of the blood pressure in the body and in the kidney. So the first function being a systemic function. This is where renin-angiotensin system, aldosterone, comes in place. So basically, it senses that there's low blood pressure in the body. So this system and cascade is sensed by this area in the kidney, and it creates renin and basically causes water reabsorption, sodium reabsorption, and this basically raises our blood pressure. This is a very important mechanism in the body, okay? The other function is the local kidney function, where we maintain homeostasis inside the kidney itself, you know, because we need to control the GFR, the glomerular filtration rate. We need to maintain this GFR so that urine keeps flowing, okay? Here it is, the juxtaglomerular apparatus. So here, as I mentioned, we have the afferent arteriole, the efferent arteriole, and then the distal convoluted tubule. Okay, can't really see the arterioles, but basically, yeah, imagine them. So here, the juxtaglomerular apparatus, we have the juxtaglomerular cells. These are the cells that are found in the afferent arteriole. So in the afferent arteriole here, we have juxtaglomerular cells present where? In the tunica media of the afferent arteriole. So arterioles have three layers, right? They have tunica intima, which is the endothelium, tunica media, and tunica externa. Tunica media is just smooth muscle cells to, you know, cause contraction and relaxation, right? In the tunica intima of the afferent arteriole specifically, there's a specialized cells called juxtaglomerular cells, and these cells, they sense low blood pressure inside the arteriole, okay? They sense low blood pressure. So what do? When there's low blood pressure, they contract to prevent more blood from going to the glomerulus and getting filtrated as urine, and like prevent the body from losing blood. So basically, they sense the pressure. Okay, these are modified smooth muscle cells in the afferent arteriole. Then we have the macula densa. From the name, macula densa means like dense. So the dense area where in the distal convoluted tubule. So the distal convoluted tubule, if you guys remember, it is made of cuboidal cells, okay? Cuboidal cells, simple cuboidal, right? However, these cells that are special, these are all simple cuboidal. These are all simple cuboidal, but what they have a special function, which is sensing the amount of sodium in urine in the distal convoluted tubule. So when there's urine passing through here, it might send there's low sodium. So what does it do? It sends a signal to juxtaglomerular cells that are right next to it, they're neighbors. And it sends the signal and tells it, okay, let's raise the body's blood pressure, let's contract, let's you know, maintain homeostasis in the body. So this is basically what they do. Okay, this is what I already mentioned. The extraglomerular mesangial cells or Lacey cells, these are pale staining cells filling the gap in the juxtaglomerular apparatus. Okay, and these are basically the signal transmission. So these are the signal transmission between the macula densa and the juxtaglomerular cells. These are sending the signals between them, telling them, hey, let's keep the body in check. Okay. So the first function of the juxtaglomerular apparatus is the RAS system, or the renin-angiotensin system, renal-angiotensin-aldosterone. Okay, so it maintains homeostasis of blood pressure. This is mostly physiology. I won't get into it, but basically what you have to know is that it maintains homeostasis, raises blood pressure when it's low, and maintains blood volume, and maintains homeostasis of sodium levels. When they're low, it causes sodium reabsorption. The second function of the juxtaglomerular apparatus is the local function at the kidney. So this juxtaglomerular apparatus keeps the kidney's pressure in check. We have the two functions, tubular glomerular feedback and myogenic function. The tubular glomerular feedback is to maintain homeostasis of GFR. When there's high GFR, it basically like lowers it. Dr. Annie didn't really get into this deeply, but basically it increases the sodium chloride at the macula densa, and the macula densa senses that, because macula densa, what does it sense? It senses sodium, okay? So it senses that there's higher sodium and causes the vascular smooth muscle to contract, and this keeps, so when the afferent arteriole contracts, there's less blood going, right, into the glomerulus, and therefore the GFR decreases or normalizes. Okay, the glomerular filtration rate. The other function is myogenic. It is to maintain the homeostasis of GFR when arterial blood pressure is high. So when there's like a lot of blood pressure at the afferent arteriole, what does it do? There's increased GFR, right? So the afferent arterial wall stretches, and the juxtaglomerular apparatus senses that. These vascular smooth muscle cells, the juxtaglomerular cells, they sense this wall stretch. So what do? They automatically cause contraction, and the GFR decreases or normalizes. These two mechanisms are renin-free. Did I mention renin at all here? No, because these are two renin-free mechanisms inside the kidney. The renin part is for the entire body to maintain homeostasis. So why is it important to regulate blood pressure? This juxtaglomerular apparatus is really, really important because it saves our lives. First of all, the filtration process of urine runs on the difference in pressure. As we know, molecules go from area of high pressure to low pressure. So it maintains the difference in pressure that allows urine to be made in the first place. Okay? There's not enough pressure, there's no urine. The second function is it prevents hypotension or low blood pressure. When we have hypotension, there's less blood flow, and tissues are not supplied with oxygen. It's really bad. Okay. Third function is maintaining the homeostasis of sodium. So the macular dense substance is low sodium. And why is sodium important? Sodium is important, first of all, because it is used in the sodium potassium pump in the body. We have all over the body, we have sodium potassium pumps on the cells, and it's also used for action potential. Sodium is part of the actual action potential in the nerves. So there's no sodium, really bad. Okay, let's zoom into the cortex. We zoom a little more. Here are the glomeruli, or the renal corpuscles. Very beautiful. You can see your afferent arteriole. Pretty. Okay. Actually, no, this is the proximal convoluted tubule. Okay. And here is the Bowman space. Very beautiful pictures. Let's zoom here. Here we have cortical labyrinth. So this is the cortex, right? We zoomed into the cortex. You see all of these renal corpuscles there. So in the cortex, we have the parenchyma, or the functional part, which are the nephrons, consisting of the renal corpuscles and the tubules and the stroma, which are the supporting cells. So if we zoom into here, we have the cortical labyrinth. Cortical labyrinth basically means maze. So it looks like a maze. Here you see all the convoluted parts of the tubules. We cut a cross section through them, you'd see a lot of circles, depending on the cut and depending on which way the tubule was moving, where we cut. We have all these different shapes of tubules, circular, straight, etc. This is the cortical labyrinth. But if you zoom into another part in the cortex also, you have medullary rays of the cortex. Let's zoom a little more. Okay, these are the medullary rays, rays like the sun rays. So here are the parts where you see, remember when I mentioned the pars recta of the convoluted tubule and the pars recta of the distal tubule? Here they are. They're erect, they're straight, and when you cut through them, they look like straight, compared to the cortical labyrinth. So, the renal cortex, you would see alternating areas of cortical labyrinths and medullary rays. Even though it's called medullary rays, we're not actually in the medulla. This is just different, confusing nomenclature, but basically this is all in the cortex. So you have alternating cortical labyrinths, mazes, and medullary rays in the cortex. And these are the renal corpuscles and the glomeruli. Okay, let's talk about the cortical lobule. I mentioned earlier, so here we have the interlobar artery, and here you would find the arcuate artery, and it branches into the interlobular arteries, okay? And these arteries branch into the afferent arterioles, okay? Now, what is a cortical lobule? What is a lobule of the kidney? A lobule is consisting of medullary ray, a line of medullary rays, which is bordered by two cortical labyrinths. So here we have a medullary ray and two cortical labyrinths. So this is one kidney lobule, okay? And yeah, medullary ray centered by, bordered by two cortical labyrinths. After we zoom, we would see the medullary rays and the cortical labyrinth. So this area is the cortical labyrinth, where it's like very squiggly and circular. Here also we have cortical labyrinth, and we have the medullary ray in the middle. Okay, this is all in the cortex. Okay, in the cortex. So let's look at the actual tubules here. First of all, this is a glomerulus, or the renal corpuscle, the glomerulus. Cool. Okay, we have, so next to it, what would we find? Proximal convoluted tubules, distal convoluted tubules. They're all like compact together. So remember the picture I showed you earlier of the nephrons, like, you know, renal corpuscle, proximal convoluted, loop of Henle. So nephrons are not actually like spread out like that. They're all compact together in real life. So you'd see all the distal convoluted tubules next to the glomerulus, the proximal convoluted tubule, they're all close to each other, okay, compact together. So here we have distal convoluted tubule, proximal convoluted tubule. How do we differentiate between them? Okay, here's my question. So this is how we're going to learn. They're very easy to differentiate. Proximal convoluted tubules are more pink and fuzzy. Fuzzy, why? Because they have microvilli. Okay, so anytime you see a pink and fuzzy, circle, because they're cut in cross-section, I mean, anytime you see like fuzziness, here also, these are proximal convoluted tubules, okay? Then distal convoluted tubules, they're more like less fuzzy, first of all, no microvilli. And another differentiating feature is proximal convoluted tubule cells, their columnar cells in these tubules are bigger. They have microvilli. And because they're bigger and there's like more cytoplasm to work with, when we come to cut the kidney slices for histology, to look under the microscope, there's a higher chance of missing the nuclei. That's why sometimes you would see a cell without a nucleus. You'd see less nuclei here. Whereas in the distal convoluted tubules, okay, there's more nuclei because the cells are smaller. There's less nuclei, there's less cytoplasm, and also the lumen is bigger because the cells are smaller. Okay, the lumen is bigger in the middle. These are distal convoluted tubule. Third differentiating feature is that proximal convoluted tubule, or proximal tubules in general, are more abundant in number. So you can see like all of these pink fuzzy areas are proximal tubules, but there's only a few distal convoluted tubules that you can see here, and maybe this is a distal convoluted tubule. You can't really tell. Okay, then we have the pars recta, or the erect part of the proximal and distal tubule. Same thing. The pars recta of the proximal convoluted tubule here, the PPR, is like the convoluted ones. The pars recta of the proximal tubule is pink and fuzzy. Pink and fuzzy. Less cells, less lumen, because the cells are bigger and they also have microvilli. The pars recta of the distal tubule, and here also it's not fuzzy, not pink, and there's more nuclei, and there's a little bit more space. You can't really see any space here. Okay, this is the medullary ray. So you see the pars recta of the proximal tubule and the pars recta of the distal tubule. Then we have the cortical collecting duct. Cortical collecting duct, how do we identify it? Like how can I tell that this is a cortical collecting duct and not a distal, pars recta of the distal tubule? Well, cortical collecting duct, if you guys remember, they are consisting of principal cells and intercalated cells. So light cells and dark cells. So here you can see all of these cells are basically very light, okay, very light cells. And you can see like maybe this is an intercalated cell, a dark cell, maybe this one, I can't find anymore. So it's very light, and the lumen is bigger. Okay, this is a cortical collecting duct, very light. Whereas even in the distal convoluted tubule, the cells are still, they have pinkiness in them. Whereas these ones are super light, the cytoplasm is white, completely white, okay? Another picture here to differentiate the pars recta of the proximal convoluted tubules and the distal, very easy to differentiate. Look here, they're pink and fuzzy, less nuclei, bigger, more abundant in number, okay? And these are more, more nuclei. You can see more bluish, more nuclei. The cells are smaller and they're less abundant. Okay. Here we have mix and match. So these are electron microscopic pictures of the cells. Here in the electron microscope, you can see how many cells? One. you can't even see the full cell in this picture. Okay, so you can identify the cell by the nucleus. So this is one cell, okay? And what do we see here? We see microvilli, and this arrow is moving from outside, from the lumen, from the microvilli to inside. So basically we have reabsorption here. So where would we find this? We would find it in the proximal convoluted tubules, and the number 4 is the pars recta of the proximal tubule. So I think you guys can understand this now. I'll give you an exercise later to like mix and match the cells just to see, to gauge your understanding. Here, this is a cell, does not have microvilli, and the arrow is showing us that the urine is moving from the capillary, or the blood, to the lumen. So there's secretion. So where would that be? That would be in number 2, which is the distal convoluted tubule, and the pars recta of the distal tubule. Okay, number 2 and 3. How do we know that this is not the cortical collecting duct? The cells are pink. They're not white. They're not pale, as in here. Here they're completely white. Okay, here they have pinkiness. Okay, here also the medullary rays, we have these cells. These cells now are completely like white. Okay, they have very little microvilli, if anything. Basically no microvilli, no organelles. See, here we saw many organelles and mitochondria, Golgi apparatus, whatever. Here there's no organelles at all. Okay, so what are these? They're completely white. These are the principal cells of the cortical collecting duct. Okay, and here we see 2 cells. Okay, and urine is moving from outside to inside. So there's reabsorption here. Okay. Okay, so I talked about this already. The cortical collecting duct is simple cuboidal. We have 2 types of cells, principal cells and intercalated cells. The arrows are showing us the intercalated or dark cells. They see there are very, very few of them. The pars recta of distal tubule here, the distal tubule, more nuclei, less pink and fuzzy. Yes, and the pars recta of the proximal tubule, large, simple cuboidal with microvilli, which give them this fuzziness, and they're pink from many mitochondria. Okay, let's play a game here. I want you guys to tell me number 1 corresponds with which cell? Okay, tell me. What do we see here? What type of cells? These tubules. Which type of cell is number 1?
The cortical collecting duct.
Yeah, so is it X, Y, or Z?
That's why.
Yes, correct. And number 2?
That's the X.
Correct, yes, because it's pink and fuzzy because of the microvilli. And 3 is obviously Z. Okay, these are the distal or pars recta of the distal tubule. Okay, here. Okay, great. Now let's talk about the renal medulla. The renal medulla consists of only straight tubules and ducts, as I mentioned earlier. Straight tubules and ducts, and straight vessels as well, which we call vasa recta. Okay, so the straight tubules and ducts that we would find in the medulla are the loops of Henle, and which makes a U-turn at the tip of the pyramid, or the medulla. We call it the pyramid. It functions as a countercurrent multiplier and creates hypertonic institution. I won't go into this, it's physiology, but it creates concentrated urine basically. And we have the collecting ducts, which merge towards the tip, and it appears to have striations on microscope because of the alternating tubules and vessels, and they're all straight. And we also have the parenchyma, the parenchyma is the uriniferous tubules and nephrons, and the stromal cells, which make up about 20% of the medulla. Stromal cells, as I mentioned earlier, are supporting cells. And these are the interstitial cells, which are fibroblast-like. They make type 3 collagen, which holds tubules, like the ones in the cortex. Okay, here's the medulla. Okay, what I need you to know from this is the cortex, obviously, right? and this is the medulla. Okay, the medulla has two parts, the outer medulla and the inner medulla. The outer medulla is consisting of the outer strip and the inner strip, and the inner medulla consists of the inner medulla, and this is the papilla of the renal medulla. You don't really have to like figure out like where the outer inner strip, inner medulla, and just know that it consists of these two parts. And then we have the minor calyx here, where this is the funnel-shaped part of the lobe, the lobe, yeah, where the urine is being excreted. Okay, so here's the renal collecting duct system. As I mentioned earlier, we have 5,000 nephrons. They merge into 7 medullary collecting ducts in the inner medulla, which eventually merge into one papillary duct of Bellini, that one straight tube. Okay, after they merge, the diameter of the epithelium of the medullary collecting duct are rising. The diameter of the epithelium is rising because we need more diameter to hold more volume because the tubes are merging. Okay, papillary collecting duct merge to give us the minor calyx, which is the funnel-shaped part that is the beginning of the ureter. And starting from the minor calyx onwards, we have 3 layers. So up until here, we've had 1 layer, simple squamous, simple cuboidal, simple columnar. However, from the minor calyx, we have, starting with the minor calyx, we have 3 layers, which is transitional epithelium. This is where the transitional or the special urothelium starts in the urinary system, which makes up the mucosa. Then we have a smooth muscle layer, which is the muscularis, and then connective tissue layer, which is the adventitia or therosa. Okay, and someone asked me, how do we differentiate between, like, are they cortical collecting ducts and the medullary collecting ducts and papillary collecting ducts the same? That the cortical collecting ducts and the medullary rays of the cortex, they have shorter epithelium for lower urine volume. Okay, and then the papillary collecting ducts of Bellini, they have taller epithelium for higher urine volume. And then when the cortical collecting ducts and the medullary they merge, they form the papillary collecting ducts of Bellini. Okay, and here, these are the points of merging of the medullary collecting ducts to form the papillary collecting duct of Bellini. Okay, let's zoom into the renal papilla. Okay, this is the renal papilla. Let's talk about all of these types of cells. So here, first of all, first thing that we can figure out is the vessels. So these are the vessels in the medulla. And what did we call the vessels in the medulla, whether they're capillaries or venules? The vasa recta, right? The vasa recta. So anytime you see vessels in the medulla, they're called vasa recta because they're straight or erect. Okay, this is just their special name, okay? But they're the same function, the same thing, okay? Then H means the Henle, loop of Henle. So we have the thin loops of Henle and the U-turn or the U-turn of the loop of Henle here, okay? And yeah, now so you might ask, how do we differentiate between the loop of Henle and the simple squamous epithelium or the endothelium of the vasa? Sorry? Sorry? You can type in the chat, okay? I can't really, your microphone's not working. Sorry. Okay, how do we differentiate between the loop of Henle and the endothelium of the vasa recta or the vessels in the medulla? Basically, the loop of Henle, you wouldn't find erythrocytes. You wouldn't find red blood cells. Okay? Easy, easy. And here, sorry? Anyway, so here we have the renal medulla. One sec. Here we have the water reabsorption by ADH. Renal medulla consists of the papillary collecting ducts of Bellini, okay? And the papilla, sorry, the renal papilla consists of the papillary collecting ducts of Bellini. In these collecting ducts, or all the collecting ducts, cortical, medullary, papillary collecting ducts, we have ADH. ADH works here. This is where water reabsorption happens. So this is where, like, basically the body sends signals if we need to reabsorb more water. This is the last site where water is reabsorbed. So the body sends signals, do we need to reabsorb more water or do we not need to absorb it? Okay, ADH helps with the water reabsorption, and also aldosterone works here on these papillary collecting ducts of Bellini. Aldosterone helps with sodium reabsorption. Do we need to reabsorb more sodium? Okay, let's reabsorb more sodium in these ducts. Okay, 4 is the vasa recta, simple squamous epithelium with erythrocytes in the middle. Okay, 2 are the loops of Henle, the thin loops of Henle. And 5 is, this is an erythrocyte, it's just like a cross section of an RBC. Okay, it's the renal papilla. Let's zoom into here. The renal papilla and the minor calyx. So this is, the renal papilla is the end of the microscopic urine flow. Okay, microscopic meaning we can't see it with our naked eye. Okay, so this is where urine filtration and excretion and formation and transport happens. Okay, it's made of simple columnar epithelium, as I told you earlier, plus microvilli. This is the last time we have urine formation. Then the area cribrosa. Area cribrosa is basically an area where we have the openings of the papillary ducts of Bellini. This is where they drain into the minor calyx lumen. Okay, and this is where macroscopic urine flow happens. Macroscopic, because we can see this with our naked eye. This is the first, like, I guess, urine transport or duct that we can see with our naked eyes, which is the minor calyx. Okay, everything else is inside the kidney parenchyma is like not visible with the naked eye. You need a microscope. So this is the minor calyx lumen. Okay, it is made. This is where we have stratified urothelium or transitional epithelium. Okay, this is where it begins. Okay, and there's no more urine formation, only excretion from here onwards. So the minor calyx, major calyx, ureters, urinary bladder, urethra, and so on. So let's zoom into here. The papilla, this is the calyx, and the transitional epithelium. So here, this is the minor calyx. Okay, this is the papilla, okay, of the papilla of the medulla, okay? These are the papillary ducts of Bellini, the papillary collecting ducts of Bellini. They're all draining into the minor calyx. This is the lumen of the minor calyx, and this is the wall of the minor calyx. Okay, this is the area cribrosa. This is just the name of the area. It's not an actual structure, it's just the area. Cribrosa because it has holes. Cribriform, it's cribriform, it has holes. So if you look at this whole thing here, okay, you would see holes because the papillary collecting ducts are draining here into the lumen of the minor calyx. And here you see the transition epithelium. Okay, we have 3 layers. Okay, we have the transitional epithelium or the mucosa, and we have the muscularis, and then we have the adventitia or cilia. So the renal papilla is simple columnar epithelium, short microvilli with the principal and the light cells. They have completely white cytoplasm. And we have the thin loops of loops of Henle that I mentioned earlier. We saw them as H, okay? And then we have the vasa recta, and yeah. Here's a slide talking about the hormones that are made in the kidney. So we have several hormones that the kidney makes. First of all, vitamin D. The kidney does not make vitamin D, it just modifies it. Okay, we have the alpha hydroxylation of vitamin D. Basically, in number 1, we have vitamin D activation. So vitamin D is activated in the kidney. So it's made in the skin, but it's activated in the kidneys. Okay, and okay, in these, where is it activated? So these are the glomeruli, right? It's activated in these tubules, which are the proximal convoluted tubules. Yeah, they're okay. Then we have renin synthesis that I mentioned earlier. Where is it made? In the juxtaglomerular cells, in the juxtaglomerular apparatus here at the vascular pole or the juxtaglomerular pyramid. Okay, and then we have here, this is number 3, we have erythropoietin hormone synthesis. Erythropoietin is the hormone that sends signals for our bone marrow to synthesize more cells, and so on. Okay, so it is, where is it made? It is made in the stratified squamous, no, in the simple squamous epithelium of the capillaries. Okay, like it's made by the peritubular, the parasites that are surrounding those capillaries. Okay, and these are the parasites that are surrounding them. Look how they're like clinging onto them. These are the parasites. They're cells that are clinging onto the capillaries. Okay, and these synthesize erythropoietin for cell production. So these are the three hormones. Okay, vitamin D activation in simple cuboid cells of the proximal tubules. Vitamin D, what does it help with? It helps with calcium absorption. Then we have renin hormone synthesis in the juxtaglomerular cells. Then we have erythropoietin producing cells, or the peritubular parasites. These are polymorphic cells. Polymorphic means they have different shapes. None of these cells look like each other. Yes, they all have feet, but none of they're not equal in shape. Okay, they have processes or feet wrapping the peritubular capillaries. They produce erythropoietin, which stimulates the bone marrow for red blood cell production. And they're located in the peritubular interstitium of the outer medulla and cortex. Okay. Okay, this is basically the last part of our crash course. Do you guys want a break, or should I continue?
I think we're fine.
You're fine. Should I continue? Okay, let's continue then. Nobody said they need a break. Okay, so here we have the excretory organs, or the macroscopic flow of urine of the urinary system. Okay, we finished from the kidney. The urine was made, the urine was transported, all that big story. Now we need to transport it from the minor calyx to the major calyx and to the outside world. So this is the cascade of how the urine flows. First of all, from the minor calyx, we have around 10 to 18 per kidney. Okay, here in this figure, you can see 1, 2, 3, 4, 5, 6, around 6, but there's 10 to 18 per kidney. We have 2 kidneys. Okay, then we have the major calyces, these ones. Okay, this is the major calyx. Okay, we have 3 to 4 per kidney. 1, 2, 3, 4. Yeah, you can see about 4 here. Okay, and then we have the renal pelvis. This is the renal pelvis where the major calyces merge. Okay, then we have the ureter. These are the ureters, and the bladder, and then the urethra, and the urine is out to see the world. Okay, they all have the same histology, which I mentioned was 3 layers. Okay, 3 layers around the lumen of urine. Mucosa, which is transitional epithelium. They all have transitional epithelium except the distal part of the urethra, which would have something else. Okay, then we have the lamina propria. So the mucosa, so we have 3 layers. Okay, we have the mucosa, the muscularis, and the adventitia or serosa. The mucosa is the epithelium, you know, and something else beneath it, which is called the lamina propria. The lamina propria is very important because it has connective tissue and the blood vessels that supply transitional epithelium. Epithelium in itself, anywhere in the body, does not inherently have blood vessels, okay? It does not have blood in it. So it relies on the oxygen transport from the lamina propria beneath it, you know, the diffused oxygen and nutrients for it to stay alive. Okay, without this, it would die basically. Then we have the muscularis, which is the second layer. It's smooth muscle. And then, and it's different. We have different like a number of muscle layers in each part I'll talk about. And then we have the adventitia or serosa, which is the outer part, you know. Okay, functions are urine storage and urine output. We don't have any urine formation or filtration here, okay? So here it is, urothelium or transitional epithelium. It is stratified cuboidal-like epithelium. It is not cuboidal epithelium. It's cuboidal-like because it looks like cuboidal epithelium, but it's not. Because why? Because it can transition into squamous-like epithelium. Okay, so that's the funky part about it. It's that it can transition from cuboidal to transitional. Okay, then we have so it's dome-shaped when it's in its cuboidal-like form. So whenever you mention this in the exam, don't say cuboidal epithelium. Just say transitional epithelium, stratified transitional epithelium, cuboidal-like. Okay, you can say cuboidal-like. Okay, you should actually depend on whether so it transitions from cuboidal to squamous-like depending on the amount of urine volume. So depending on the amount, you write cuboidal-like or squamous-like, okay, in the exam. But don't write cuboidal, write cuboidal-like. Then these have round nuclei in the top layer. That's why they're cuboidal-like, because we classify epithelium if it's cuboidal, squamous, or columnar based on the layer on the top. Okay, if the layer on top is cuboidal, it's cuboidal. If it's the nuclei are flat, if the nuclei are flat, it's squamous. Okay, and so on. The minor calyx has 2 layers. The major calyx has 4 to 5 layers. I'll talk about those later. And they have an impenetrable barrier of urine with tight junctions. The function of transitional epithelium is that it adjusts to low and high urine volumes. So when there's a high urine volume, it changes shape, okay? So depending on high or low urine volume, it increases the number of layers or it decreases the number of layers to make it thinner. And it also changes the area of the plasma membrane. It can invaginate onto itself, which I'll show you what it looks like later. And this is the urinary excretion system. Sorry, this is a typo, excretion. Okay, here's a picture. Okay, a slide. So here, tissue is not stretched, and here tissue is stretched. When it's not stretched, basically this means that there's very low urine volume in wherever we are, okay? So when there's low urine volume, the layers are higher, the lumen is smaller, okay? We have around like 6 to 8 layers here and here, okay? This is what it looks like. We have dome-shaped, okay? Here the urine, okay? But when the tissue is stretched, when it has to accommodate more urine, what does it do? The layers become like flattened, they like spread out. We have 2 layers now, and the plasma membranes invaginate. I'll show you how. Okay, here, is this where, do we have high or low urine? Okay, it says low urine and lumen. Okay, they're dome-shaped, okay, cuboidal-like epithelium. Okay, here's how they invaginate. So this is the special thing about transitional epithelium. Okay, so Dr. Annie put this picture of an accordion. I think it's a perfect, analogy as to the plasma membrane and transitional epithelium. So basically what happens is that these transitional epithelial cells, which are dome-like, when there's high volume, when they're low volume, they're cuboidal, they're holding the volume, okay? But when there's high volume, they need to make more space, okay? So they open up, okay? But then when there's like low urine volume, what do? Where does all this plasma membrane go when you're squeezing them? It invaginates onto itself, invaginates means it goes in on itself, and it creates these plasma membrane invaginations, which are called vesicles, okay? They're not actual vesicles, because we know vesicles are like organelles and inside the cell, but this is a misnomer. They used to think these are vesicles, these are like organelles and like specialized organelles, but no, this is actually the plasma membrane itself folding in on itself and creating these invaginations. So when the plasma membrane, when you close this accordion, what happens? It becomes smaller, okay? When it opens, the plasma membrane invaginations, we have more surface area and they can hold more urine volume. Does that make sense?
Yes.
Okay. Here's another picture where you can see the vesicles, okay? And here in this picture, we can see 2 cells, okay? Plasma membrane. All Dr. Annie needs you to know from this picture is that there are tight junctions between them, okay? There are tight junctions between the cells. That's it. And these are the invaginations or the vesicles or the plasma membrane going in. Okay, this is a slide of the ureter. Do you think there's high urine volume, or there's like a lot of urine here, or not much urine? Little urine.
A low urine volume.
Yeah, it's a low urine volume. So because it looks like a star shape, okay, it's like the ureter is not stretched out. Okay, when it's stretched out, it would be like a circle. Okay, this is the lumen where urine flows. This is the ureter. This is a transitional epithelium in the ureter. Here we have the F. The F is, what is the F? Is the lamina propria. Lamina propria is connective tissue plus the blood vessels that supply this transitional epithelium, okay? Then we have the muscularis, which is the muscle layer, and the adventitia, okay, or the serosa on the outside. Okay, so here's in word form. Okay, the mucosa is transitional epithelium, which is stratified cuboidal-like, plus the lamina propria. And then we have the muscularis layer, where we have the proximal 2/3 of the ureter are two layers, like the calyx, which is the inner longitudinal, and the outer circular. So remember as like LC or LCL. The proximal 2/3 of the ureter is LC, and the distal two parts is three layers, inner longitudinal, outer circular, and outermost longitudinal, LCL. Okay, LC, LCL, the distal 1/3. Then we have the adventitia surrounding it. Because ureters are retroperitoneal or behind the retroperitoneum in our abdomen, they are surrounded from the like anteriorly, they're surrounded by the mesothelium of the peritoneum. Posteriorly, it's surrounded by connective tissue, and there's adipose tissue in between. Okay, the function of the ureter is peristalsis of urine as bolus because it has the muscular layers, the LC and LCL. Okay, urine does not flow continuously; it moves in boluses or like in like groups. Okay, like a bolus of urine, a group of urine. So it's squeezing urine from the kidney to the urinary bladder. Okay. Okay, here's like a Photoshop picture of a kidney stone. If we had a kidney stone in the ureter, and it scratched the epithelium, would there be blood in the urine? What do you think? Okay, there would be no blood. Why? Because transitional epithelium or any epithelium inherently does not have blood vessels. It receives its blood from the lamina propria or, the connective tissue underneath it. So if it only scratches the transitional epithelium, we wouldn't have blood. But if it like scratches all of that and then reaches here, yes, there would be blood in the urine. Okay? Okay. Let's talk about the urinary bladder. This is the urinary bladder, the urinary vesicle, the urinary cystus, whatever you want to call it. But we call it the urinary bladder because there's another bladder in the body, which is the gallbladder. Anyway, the urinary bladder this is what it looks like under the microscope. So we have the serosa or adventitia, the muscularis, and the lamina propria, and the transitional epithelium. Okay, the transitional epithelium here, we have rugae, or they're like folds, okay? And they're actually visible by the naked eye, okay? And these folds are basically when there's a high urine volume, these folds like flatten and the surface area increases to hold more urine, okay? These are rugae. Okay, the structure of the urinary bladder, we have the mucosa, which is folded into rugae, and which it's made of transitional epithelium and lamina propria. And it is dome-shaped when the bladder is not distended. Binucleated. We have two nuclei in this transitional epithelium. Then we have the muscularis layer. Like the lower part of the ureter, we have LCL, inner longitudinal, outer circular, and outermost longitudinal. Then the adventitia, because it's infraperitoneal, which means the peritoneum is above it. We have superiorly, it's surrounded by mesothelium of the peritoneum, and there's connective tissue where there's no serosa. Okay, the function of the urinary bladder is distension as a reservoir of urine. So there's no rugae when it's distended, they flatten out. And contraction during micturition. Micturition is peeing, okay? And it relaxes during urine storage, rugae present. Okay, let's zoom into the rugae here. Okay, this is the transitional epithelium here. I told you there are two nuclei, it's binucleated, which is pretty cool. And they're dome-shaped. So basically there's low urine volume right now. This is a trigon. All you need to know about it is that, so in the entire bladder, we have rugae, right? Which can stretch when there's high urine volume, except for the trigon. This is the area that does not have rugae. And what does it do so can you imagine when there's no rugae and there's a lot of urine, okay, and stretching, and there's no there's no like mechanism for it to stretch. There's no like, projections for that are able to stretch. So what happens? It senses that there's a lot of stretching happening, okay? So what does it do? It sends a signal to the brain, telling the brain, hey, like I'm stretching. There's a lot of urine, time to go to the bathroom. Okay, this is the trigon. Because there are no rugae, it senses the high pressure, whereas the rest of the bladder has rugae. Okay. Male urethra, let's talk about the urethra. So the male urethra consists of three parts: the prostatic, the membranous, and the spongy. The prostatic urethra is where the transitional epithelium ends. Here we see the last transitional epithelium in the body ever. Sorry. So in the prostatic urethra, you have transitional epithelium, okay, dome-shaped, cool. The second part is the membranous urethra and the urogenital diaphragm. This red line is basically the muscle of your pelvis, I guess, that's holding your entire, abdomen together. This is the pelvis, and in this, in between these muscles, we have the membranous urethra. Okay, that's passing in between it, and the membranous urethra is stratified columnar epithelium. Here, stratified columnar. We don't have transitional. We have stratified columnar. Sorry. Stratified columnar or pseudostratified columnar epithelium. Okay. Then we have the spongy urethra. The spongy urethra is the part of the urethra that is in the corpus spongiosum of the penis. It has two parts. So the entire thing is stratified columnar or pseudostratified columnar. Then in the external urethral orifice, we have the epithelium here. The more proximal epithelium, we have stratified squamous. Okay, we have stratified squamous, which is non-keratinized. The outer part of the external urethral orifice, which is the skin, basically, it is stratified squamous keratinized epithelium. Then the female urethra has two regions, which is the proximal and the distal. The proximal part of the urethra is where the transitional epithelium ends. So here, the transitional epithelium ends in the prostatic urethra, and here the transitional epithelium ends in the females in the proximal urethra. So the first part of the urethra, transitional epithelium or urothelium. The middle part is stratified columnar or pseudostratified, just like the male urethra. And the last part, or the external urethra, orifice or meatus, we have stratified squamous, non-keratinized. And when it goes into the skin, the last part, which is like the skin part, basically, it's keratinized epithelium. Stratified squamous keratinized. This is just a slide of the prostate. Okay, these are the acini and all the glands where like the prostatic secretions are made, and this is just the fibromuscular tissue surrounding it. You don't need to know much other than that, but this is in a young male, okay, in a young person. Then in an old person or older male, you would have these, which are like prostatic, you could call them like prostatic crystals or prostatic stones. Okay, they're benign. They don't really do much. You would find them in a male prostate. Okay, that's the only difference. This is just a summary slide. Let's talk about the sphincters. The sphincters in the male, we have the internal urethral sphincter and the external urethral. The internal urethral sphincter is voluntary or involuntary. It is involuntary because it is part of the detrusor muscle of the bladder, okay? The bladder, by the way, I forgot to mention the bladder, yes, it's three parts: inner longitudinal, outer circular, and outermost longitudinal. This whole part, this whole like muscularis layer is called the detrusor muscle. It has a special name, the detrusor muscle in the bladder. The detrusor muscle, a part of it, makes the internal urethral sphincter, okay, which holds urine, okay? So it is involuntary, okay? And it's controlled by the autonomic nervous system. The outer or external urethral sphincter it is controlled by the urogenital diaphragm, okay? And it is voluntary. So when we teach a kid how to do potty, okay, potty training, Are we teaching them to control this sphincter or this one? The internal or the external sphincter?
The external.
Yes, external, because it is voluntary, controlled by the somatic nervous system. So we teach them how to control the external urethral sphincter in order to hold urine. In the female, we have the same. The internal urethral sphincter is part of the detrusor muscle, and it is involuntary, controlled by the autonomic nervous system, and the external is voluntary, controlled by the somatic nervous system. This is just the cool thing about dialysis. This person's kidneys are failing. They don't work anymore. So this is basically his kidney. It filters urine and gives it to him and gives it back. It takes his urine, gives it back. This is his kidney. Okay. This is a renal biopsy where basically we take a tissue from the kidney and we examine it under the microscope or the light or the electron microscope to see if there are any abnormalities. And yeah. This is a glomerular crescent. So this is a normal glomerulus, right? You have the Bowman's capsule. When you have certain diseases happen to the kidney, you have a glomerular crescent, which is like an overgrowth of some cells. Do you think this is normal that there's like cells here in the Bowman's capsule?
No, it's not.
Yeah, it's not normal. It's not okay. Okay, this is like, it leads to diseases and nephrotic syndrome that I won't get into. Then we have kidney stones. Kidney stones, you can see here, it's like white appearing on the image. And all you have to know about this is that when there's a kidney stone, you would have a dilated ureter, as you can see here, it's very visible, and a bigger kidney, dilated calyces, major minor calyces. This is bad because it causes hydronephrosis or like a lot of urine or volume in the kidney. This is a normal kidney, normal calyces, normal kidney. This is how they should look. When they're dilated, the urine is not flowing. So when they dilate and the urine is stagnant or staying inside the kidney, this would cause infections and so on. And as you can see here, the bladder would be very flattened, okay, because it's not filling with urine. The calculus or the stone is preventing it from going down, preventing the urine. All right. And we're done. Let's have some questions. Can anyone read this question and answer it? Pretty easy. So can you tell me the answer in the chat? It is B, the nephron. Which of the following terms best describes the basic structural and functional unit of the kidney that filters blood and produces urine? It is the nephron. The nephron is the functional unit of the kidney. This is these are the structures that are doing the function of the kidney, which is urine formation and filtration and so on. Okay, a section of the kidney is obtained at the biopsy examined by light microscopy, shown in the image. Identify the zone or region indicated by the double arrow. What is this zone of the kidney? Are you writing in the chat? I'm not seeing. Oh, B. Yes, okay. 67-year-old man is found to have blood in his urine during a routine checkup. A CT scan reveals a renal mass that is subsequently removed. Microscopic examination of the surgical specimen reveals normal tissue along the tumor margin. So this is a normal tissue shown in the image. Identify the region or zone indicated by the double arrow. What is this region called? Yes, it's the medulla. Okay, because this is the papilla of the medulla. Okay, for the surgical specimen shown in the previous question, identify the open space indicated by the asterisk. What is this open space? Funnel shaped. Minor calyx, yes. A kidney biopsy is embedded in paraffin section stained with hematoxin eosin and examined by light microscopy. Identify the structure within the oval. The structure within the oval, this one. See? Yes, it's the macula densa of the distal convoluted tubule. Okay, this distal convoluted tubule is the dense part of it. That is the macula densa. Okay. Which of the following biological processes describe the critical function of the structure described in the previous question? B. Yes, monitoring sodium in primary urine. Which and the juxtaglomerular cells, they monitor the blood pressure in the afferent arteriole. Several retroperitoneal organs are examined at autopsy. One of these organs is shown in the image. The histologic features of the specimen suggest that it was obtained from which of the following anatomic locations? Yeah, this is the ureter. The apex of the medullary pyramid is examined in the histology lab. Identify the tubular structures indicated by the asterisk. Yes, these are the papillary ducts of the Bellini. They're draining into the minor calyx. Which of the following biological processes describes the primary function of mesangial cells? Yes, it's C, phagocytosis and endocytosis. And here are some important points that you can read later, but if she asks about the type of cells, you say columnar, cuboidal, squamous, field cells. If she asks about the type of tissue, you say stratified or simple. And whenever the doctor asks you to name a cell in the collecting tubule, as I mentioned earlier, say principal or intercalated, not dark or light. And sphincter smooth muscles never hang out alone. They are always part of a muscle layer. They are thickening of the circular smooth muscle layer. So on the internal urethral sphincter, it was a thickening of the detrusor muscle of the bladder. And the external urethral sphincter was part of the urogenital diaphragm at the bottom. And here are my references. And thank you. Do you have any questions? Thank you. Does anyone have any questions? You can open your mic. Thanks a bunch. Of course. I'll wait for questions if anyone has any, but if nobody has any questions, I guess that's it. This is a feedback form if you guys want to give me feedback. And thank you guys so much. Have a good evening. Good luck on your exam.